This is episode 94 of the Immunology Podcast, Engineering Immunity with Dr. Lili Yang. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Lili Yang from the University of California, Los Angeles on the podcast to talk about her research developing new immunotherapies for cancer. We've also got our usual roundup of recent highlights in immunology news coming up, but first...
Registration for Immunology 2025 opens on December 10th. Connect and collaborate in Honolulu, Hawaii from May 3rd through 7th, 2025. Advance your career and access cutting-edge research. There has never been a more important time for the immunology community to gather at the world's leading annual all-immunology meeting. Learn more at immunology2025.aai.org.
All right. Well, you just got back from holiday, right, Brenda?
I did, Jason. It's been a long time.
I'm jealous of your holidays and will remain so forever.
That's okay. I'm fine with that. Yeah, it's been nice. So for listeners who didn't hear me gloat about it on the previous episode, I just got back from a trip to Argentina, to my home country, to my motherland, to the land of tango, dulce de leche, and very, very bad economic policies. So yeah, that was fun.
Did you participate in tango, dulce de leche, or bad economic policies?
Definitely dulce de leche. That was the first thing I did. And as soon as I got out of the plane, it's just stuffed my face on that sweet, sweet thing. For those who don't know, it's kind of milky caramel that is extremely common. It's like the peanut butter of the US in the US, but like sweet and definitely not healthy at all. But man, it's good to have a spoonful of it when you get back home.
Interesting. All right. But no tango.
No tango. No. I spent my time traveling a bit. We went to visit some of the tourist nature attractions in the northwest of the country, near the high mountains. I spent some time at a couple thousand meters above sea level, which was fun, and visited some of the very nice landscapes, kind of lunar landscapes. Very, very pretty. Really pretty place to visit. I hadn't been before. So it's nice to get to know your own country when you're living abroad.
Any altitude sickness?
No, I was prepared. I was a little bit scared. Some people tell me they get this whole awful headaches and they kind of walk. And to be honest, I felt fine. The only time I did notice we were at 4,000 meters above sea level is when we had to do a little run back from escaping from a storm coming with, some nice lightning. And we thought, well, this is not very safe. So let's just go back to the car. And we had to kind of run a little bit uphill. And at that point, I realized, yeah, I am kind of high and my legs are not responding normally.
A little bit of burn.
It was a little bit tough. I was like, oh my God, I'm running out of air. This is tough. But otherwise, I feel fine. So I feel, I did grow up at, 720 meters. So I guess that gave me a little bit of a head start. I don't know. All right. So what you got? So long. What's the science today?
We're starting with a little apoptosis and pyroptosis.
Ooh.
And what the mitochondria are doing. So a little immunometabolism. So this came out in Immunity on November 20th. Its title's Acute Suppression of Mitochondrial ATP Production Prevents Apoptosis, Provides an Essential Signal for NLRP3 Inflammasome Activation. First author is Benedict Saller. Last author is Olaf Grob, but that's a beta, not a B, and I don't know how to say that.
Ross. It's like the double S in German. I guess it's a German.
Ross. Olaf Gross. All right. It is Germany. There's like 34 subnotes for the various authors and institutions.
Collaborative paper. Here we go.
Yeah, here we go. All right. So This is one of those papers where there's a lot more there that I will lose in describing if I try to describe every step of a signaling pathway and every molecule that they did. But I think the high level is very interesting to everyone. So there's something that we know where if you have like NLRP3, inflammasome activation, you get pyroptosis, right? So pore formation, boom. But there's this thing where you have more pyroptosis and you have less apoptosis. And so they're trying to figure out what was happening there. And they did a lot of this with pharmacological agents that would either activate one or inhibit one of these two pathways between apoptosis and pyroptosis across various pathways. And so what they found, and this all started with this observation that nigericin, they showed first that rapthanol induced, which is an apoptosis inducer, and nigericin, an LRP3 activator, combined that causes mitochondrial apoptosis and activation of LRP3. What interesting but the nigericin co-treatment blocked the apoptosis through caspase-3. And I'm like, well, that's weird. Why is this happening? And so that's the root of this paper. And what they first find is that this inhibitor with nigericin, it works by blocking ion efflux, which they actually show a little further down, which is already known. But what they find is that it decreases ATP production by blocking the ion influx, right? So you have less F0, F1 ATPase activity. And that then traps cytochrome C between the membranes. And so they identify this through EM, and they look and see membrane structure changes for the mitochondria. And with that cytochrome C release, that is blocking apoptosis. But they show that this is a universal process. So NLRP activators, which block ATP production, do this. It's a generalized mechanism. Oxidative phosphorylation inhibitors, which are again going to drop ATP production, do this. So if you have a chemical or viral process, because they also identify that infections like SARS-CoV-2 and influenza, which honestly, they threw this on as a very last thing with one last figure of the paper, I think, to give some biological relevance. Of course, they show that those also block APT production and inhibit apoptosis in favor of pyroptosis. But decreased ATP production starts the cascade in the mitochondria for NLRP3 activation, so inflammasome activation, but it's insufficient. And so you have to have other signals. And so they show it through things like the imiquimod system, inhibitors or drugs, and Yoda-1, which is just a hilarious name for a drug. But they couldn't identify the signal driving that. But they did show that while you have this central axis of decreased ATP production, which is what NLRP activators do, that is not sufficient for pyroptosis, but is sufficient for inhibition of apoptosis. And so they're really trying to do, and this is one of those, how does the immune response get influenced, and why does inflammation for inflammasomes then block apoptosis, right? So why do you get pyroptosis, not apoptosis? And it's really showing that the inflammatory response works to decrease the mitochondrial release of cytochrome C. And that stops the whole thing for apoptosis happening. So that's the big point of this paper, and everything else, depending on where you are in your life as a scientist, there may be a lot more there for you in terms of you need to know about the signaling. They go into a lot of depth, but for the high level, it's important to know that inflammasome activation blocks apoptosis, or at least intrinsic apoptosis.
We discussed kind of this idea, right, of, I think with some of our guests or in previous papers, that this idea that if you block apoptosis, you actually get this pro-inflammatory death. We discussed it with one of our guests very recently. So it's nice to see that this is an understanding of the mechanism behind that, right?
Exactly. They're starting to see some of the mechanism behind it and how the pathways are happening.
Okay. And in the end, it's all about the mitochondria getting all worked up, right?
Yep. Or it's all about the mitochondria having some stuff happen to them. The central role of the mitochondria.
Right. I mean, powerhouse of the cell seems like what would be a limiting description of this organelle by now. I think we should definitely stop saying that.
There's so much more than organelle. There are little bacterial symbiotes coming to live with us forever.
And yeah, and providing us information apparently too, and just like telling cells to getting cells to die and things like that. I don't know. Sometimes they're I think they have too much power. You should get I don't know.
To liberate ourselves from the mitochondria. Got it.
I'm not sure I'm comfortable with that. All right. I mean, why are they overriding the nucleus instructions and getting cells to die? I'm not sure I like that. All right. So talking about cells dying, uh, I was gonna present this paper second, but I'm gonna start with this one just because we're talking about inflammation or cells dying and bad things happening, and you mentioned SARS-CoV-2. So you know what? I figured I'd continue. I'm going to just briefly talk about a paper that came out officially on Cell Host and Microbe, uh, technically on December 11th. So that is in the future. But it's just, the publication date. It's already been around, or also as a preprint, and it kind of caught my attention because it got some traction on Twitter and a little bit of controversy, people taking things from the paper out of context, people getting angry about that. So I figure, let's just dive into the controversy and just maybe discuss this paper and see if there's any merit to what people have been talking about. So the paper is called Persistence of Spike Protein at the Skull Meninges Brain Axis May Contribute to the Neurological Sequelae of COVID-19. And it has, again, like you mentioned, I think another reason why I figure continue with this paper, a lot of authors. But for what I gather, there are four first authors sharing that title. Shou Shui Rong, Han Cheng Mai. I'm sorry, guys, if I'm butchering your names. Gregor Ebert and Saket Kapoor. They are all first authors, and the corresponding author is Ali Erturk from the Ludwig Maximilians University in Munich. That's in Munich, in Germany. And the reason why this paper got my attention is because basically they have this kind of fancy protocol in which they can do full tissue stainings. And they kind of clear the tissue and stain it, and this allows them to see things in full kind of biopsies and also like full mouse bodies. And they use this tool to look into spike protein distribution in mice and in human samples from brain. And they kind of, to some extent, conclude that they can show that there is persistence of spike after uh, COVID, in even in people that don't have acute COVID. And then they do a few studies in which they inject the S1 subunit of spike in mice, and they show that it gets distributed across the mouse. So let me just go a little bit over this. Of course, one of the main questions now is we know this post-COVID, this long COVID syndrome that happens um, in many people that were infected with the virus. Much of it has some kind of neurological aspect to it, and many people are suffering long-term, problems with cognitive problems and anxiety, some behavior problems, or so. It's really hard, I think, nowadays to there's so much controversy about everything around this syndrome, but I think we can say that definitely it's something that exists, that people are suffering the effects of that COVID infection that happened a long time ago, and they're still having cognitive issues because of that. So the question is, where are these issues coming from, and why didn't we really see that with other viruses? I mean, we've had other, severe viral infections in the past. I think in particular influenza, for example, and then they use influenza in this paper as kind of a comparison. But is there something special about COVID and the proteins that COVID has that is having a outsized effect on our neurological, health? And so what they do is in this paper is that they start with samples from patients they have this postmortem skull tissue from patients that died from acute COVID. And this is important, right? Because these people, they if they die from acute COVID, of course, they're gonna have a lot of virus because that's probably what killed them. Um, and they see, of course, when they look into they stain, the antibodies against spike. I think this case is so they have an antibody can detect spike. I'm not sure if it detects the S1 or just other part of the spike protein, but they show, of course, that they detect the spike protein in the samples of brain of patients, and they see it outside of the blood vessels. So when you think about, if you think the blood vessels have these ACE2 receptors, so I guess it's not so strange that there would be viral infection there, but they detect the spike protein outside of the blood vessels, and this suggests that at least the protein extravasated from the vessel into the skull uh, marrow tissue, because they're looking inside there's this bone marrow inside of the skull that you can find in humans. So I guess that it's not so surprising that you find um, spike protein in patients that died of COVID. I think the point that they want to make is that they can see this kind of leaving the vessels into the brain parenchyma. And I think that's kind of the point they want to make, that the spike has access to the brain itself through microhemorrhages or things like that, that will bleed into the brain. And I think the thing they want to make is that they also had samples that were technically, so the patients that did not die of COVID, or at least not, that was not the cause of death, and technically they were PCR negative, so they didn't have an ongoing viral infection, and they also can detect spike protein and nucleocapsid protein in several of the samples that it got from these patients. And this is kind of making the point that the spike protein seems to outlive the infection and persist longer. Uh, for It's hard to say because I don't have the information of how long these people, like when was the last COVID infection in this patient. So it could have been months, it could have been last week. It's a little bit difficult. But in principle, you can have spike protein in the absence of an ongoing infection, and that you can find it in the meninges and in the skull. So making the point that, well, they can have this inflammation caused by so this protein is still there. And they so this kind of the idea that you can have this persistence. And they have this mouse model in which they have the ACE2 protein, so this receptor that it's targeted by SARS-CoV-2, which is most SARS-CoV-2 forms only recognize the human version of this ACE2 receptor. So what they do, they have this transgenic mice in which they have the human ACE2 receptor expressed under the human keratin 18 promoter, which, for what I understand, gets also expressed in epithelial cells in mice. So basically, you have this kind of model of ACE2 expression in mice. And here it gets a little bit complicated. So they infect the mice, and they basically see viral expression all over the mice. And I guess it's a little bit difficult to say because this is a non-physiological expression of these receptors. So people are like, I was a little bit unsure. They have a different system in which they are uh, injecting the spike protein itself, and then they see that they have it gets also really distributed across the mice a lot. And again, this was a little bit so basically they show that it compared to, for example, proteins from influenza, that this distribution seems to be much more kind of far reaching across the mouse. And this kind of makes the point that uh, they point that the spike protein is able to really distribute itself across the mouse and potentially across a human subject, which is difficult to say. You cannot analyze every single tissue in a human. Um, and they also use a different spike version, which is has a mutation that allows it to bind to the mouse version of the ACE2. And this is the one that you use a lot to show distribution of spike protein across the mouse. If they inject the mice with spike, this version of spike, it gets distributed across organs, including kind of the blood-brain barrier and potentially the brain of the mice. The point, so basically what they want to show is that spike is different from other proteins and that it can apparently reach other targets. If you have a severe infection, you can get that spike in all over at least in this mice, and that it's just having the protein itself can trigger new inflammation and can trigger changes in protein expression that are associated with inflammation, and that this on itself can be, at least to some extent, mediating the inflammation that you see. And because spike protein can persist in time more than the virus itself, this might be real. So one of the points that is made, or like people are making, is that this can be related to long COVID and the neurological component of long COVID. And I think that's kind of the main. I don't want to go too long because there's a lot of other experiments, but I think they make some analysis in which if they inject spike protein alone, they get some kind of behavioral changes in this mice, and that is kind of corresponding to this anxiety behavioral analysis that they do in mice. And there's some changes that is just by injecting the spike protein alone. And they make the point that spike can indeed interact directly with some proteins. I think, for example, we have some study that we shared before in which they can interact directly with fibrinogen and make these fibrin clots on itself, just because of the kind of the structure of the protein itself. And basically, the idea is that you can have this persistent spike can generate or exacerbate neurological damage. They have some models in which they do either traumatic brain injury or a stroke model in mice, and they show that these mice that have this accumulation of spike, they respond worse or they recover worse from this specific neurological insults. And I think that the last point that they make is that if they vaccinate the mice with mRNA vaccine before, and then they infect them with an Omicron variant, that they observe lower spike protein in all of the organs that they analyzed initially, including the skull marrow, and the brain. I find that particular thing not so surprising, because if you have a vaccine that protects you against viral load, then it also makes sense that you're going to have less of this thing circulating and therefore less of it accumulating in tissue. So that particular thing, I don't know, I thought it would make sense. But in general, I think it was an interesting paper. It was interesting to see kind of the whole body stains and look, but I don't think it is necessarily very, I don't know. I think the kind of conclusions you can make from this paper are limited. So a lot of controversy or other people making big claims from this paper, I think, is not necessarily called for. I think in general, what this paper shows is that you can have spike after what they see in humans. There's still spike protein after apparently having cleared the infection. And I guess the idea that spike seems to be on itself, alone, immunomodulatory or immune inflammatory. Maybe that I think that could be the case. But I don't think this necessarily is going to explain long COVID as things are right now. But I don't know, everything we know, I think is important to know. It would have been nice to know whether, we could, these people, the kind of the long COVID patients that had spike, how long ago had they had a COVID for the last time, and whether they actually had symptoms of long COVID if they found spike in their brains.
Well, that's my question. You said these people who they had the brain samples from died of COVID, so they had to have had it actively then.
So they have two sets. So the first set was people that died of COVID.
Okay.
So then you would say, well, I'm not so surprised they had spike in their brain. I mean, I think.
Well, sure. Yeah.
And then they had a subset or some samples of patients that had not died of COVID, but they examined skulls of patients who died from other diseases in 2021 and 2022. And of the 34 samples that they analyzed, they saw spike protein staining in 10 of them.
So how do we know that's not just asymptomatic infection?
That's the thing.
I mean, because that's what was happening. People were going to the hospital with all this other stuff found to be COVID positive, weren't just asymptomatically infected.
Sorry, I had to make a small correction here because I think I misunderstood something. They had the patients, they died of COVID. Mm-hmm. So they show that not all of them. So in the samples that were in the brain cortex samples, they were PCR negative. So in these brain cortex or in the samples from the brain, none of them actually had PCR. So they were all PCR negative, but they still had the spike. So they
died of COVID.
They died of COVID. So that's why I thought it was PCR negative, but I don't see any PCR data on people that die of non-COVID causes. So actually, I don't know if they have guaranteed negativity.
All right. Well, I think we may have to keep moving on here.
Yes.
So I'm going to do a blast from the past, like COVID's a blast from the past. One of our first guests has a paper that just came out in Cell Reports. It is Combinatorial Genetic Engineering Strategy for Immune Protection of Stem Cell-Derived Beta Cells by Chimeric Antigen Receptor Regulatory T Cells. November 26th. First authors are Jesse Barra and Rob A. Rubino. Last authors are Holger A. Russ and our old friend Leonardo Ferreira. All right, so I'm going to keep this real quick because I don't think you have to talk very long to talk about why this is so cool. So what they did is they created a chimeric antigen Treg, and they did this in mice, but had a specific bait receptor on it that was immunologically inert, that was a truncated epidermal growth factor receptor, known to be biologically inert, and that was what was used for the CAR-Treg homing. All right, and then so they combine this with human-derived, pluripotent stem cell that were pancreatic beta-like cells that had this EGFR truncation on it. And then if you put those cells on their own in a mouse, right? So these are beta cells that'll produce insulin. But if you put these in a mouse normally, the mouse's immune system kills them over time, right? And so you see this in the paper. If you combine it with the CAR-Treg that's specifically targeted, they don't die anymore. And these are human cells in a mouse. So you can imagine you're going to be even more successful if you put human cells in a human, or if you put human cells in a human that were genetically matched, because you can derive beta cells from the host as well. And so the notion is, and they've been having trouble getting pancreatic transplant beta cells to work for treating type 1 diabetes, because your immune system hates your beta cells and keeps killing it over and over again, right? But if you create this chimeric beta cell with this bait, and then a CAR-Treg that targets that bait, you get protection of these transplanted HPSC-derived beta cells. And that's the entire paper. I mean, it takes five figures to get there. But that's basically what they did. They basically created a beta islet cell transplant that, with the CAR-Treg on top with this specific unique bait, which is not immunologically active, it's an inert protein, the cells survive. And then they show that, and then they drop the mic and walk away. The only reason it's not in the bigger paper is it's not in humans.
Pretty neat. So they make these beta cells, they just have their own bodyguard, so to
They
say.
make a beta cell, human-derived pluripotent stem cell beta cell with this gene added in, and then a CAR T reg that targets that gene, that protein, right? And then because of that, it saves the cells. But it's a T reg specific to those cells, so it's not having T reg activity everywhere.
Right. What is the protein that they use again?
It is human epidermal growth. It's a truncated human epidermal growth factor.
Okay. Okay. Very commonly used as a marker.
Yeah.
So I guess it's something I start. That's pretty cool. I like it.
I don't have much more to say other than that it was awesome, and it's really cool. It'd be really cool to see this translate.
Yeah, for sure. So that's okay. Okay. Nice and short and sweet. I like it.
And CAR
Okay.
T reg and some diabetes all in one.
All in one. All right. I'm going to try to, because we're running out of time. So I'm going to try to keep the second one short. I think I rambled too much about the first one. I just want to say to the authors of the paper, I'm sorry if I sounded skeptical, but I think it's pretty nice work, but it's hard to, translate these studies to kind of clinical relevance. But anyway, moving on. Also, from a ghost from the past, we have a paper from the lab of Akiko Iwasaki and first author uh, Yang. And the paper is published in Science Immunology and is called A B cell screen against endogenous retroviruses identifies glycan reactive IgM that recognizes a broad array of enveloped viruses. And just to keep it short, ERVs, that is endogenous retroviruses, are the sequences that are all over our genome. They're remnants of previous infections by retroviruses and their integration in our genomes. And they have been happening for millions and millions of years. And apparently, about almost 10%, up to 10% of our genome is of mice and men is composed of these truncated sequences. So it's like a lot. And most of these loci, most of these genes are not functional. So they're just there. But we do have a few. So mice and people also have a few kind of copies are capable of translating. So they have like the coding sequence is still conserved. And this is particularly kind of observed in mice. And I just want to say that so there's different types of ERVs, but let's just focus on the one that is kind of discussed in this paper. In certain mouse strains, if you have deficiencies in B cells or TLRs, particularly toll-like receptor 7 signaling, it happens that there's this emergence of infectious ERVs, particles from black 6 mice, for example. There's in the case they mentioned here in black 6 mice, there's a single locus called EMV2 that has the capacity to generate infectious variants if you kind of shuffle the thing enough to be kind of expressed. So it can recombine with other loci, and then you can have kind of restore the viremia of this virus. So there's like this latent, this kind of little bomb waiting in the genome of these mice, and if you kind of cross them, and if you give them the right background without this gene, without this kind of immune surveillance, you can have actual virus production. And what's interesting is that this is the absence of B cells, and toll-like receptor 7 seems to be initiate this activation. So these does look like these B cells are providing some protection, and it seems to be possible through a T cell independent B cell response. And I'll get back to that in a second. And also in mice, they can still get protected. Mice that are incapable of undergoing germline terminal hypermutation. So it does seem that there's some protection kind of encoded in the germline, in the German sequence of B cell receptors or B cell or antibodies. So there's a subset of B cells known as innate like B1 cells that I actually wasn't very kind of aware of. I had heard of it. I was like, but it's just really cool because B1 cells are the counterpart to the classical B2 cell type, which are just the regular B cells as you know them. You know, they come from the bone marrow. They get active. They if they find their antigen, they get activated and they collaborate with T cells to get to change their hypermutate their chains. I know increase their affinity and do germinal centers and blah blah. But there's this other subset of B cells, these B1 cells. They're actually of fetal level origin. They don't interact with. They're not depending on T cells for their activation. They mostly generate high quantities of IgM and apparently also IG3. I don't know how that works, but basically they don't hypermutate their sequences, so they only use germline encoded things after the initial, recombination. That's it, and they are known to recognize kind of some conserved pathogen antigens. So that's why they are kind of called innate like, because they are kind of a pre-existing repertoire capable of recognizing sequences that are commonly found on pathogens. So what kind of this paper talk, I want to just kind of go a little bit to the point of the paper. They show that there is a population of these B1 innate like B cells that are already expressing antibodies that can recognize glycoproteins that are encoded in this retroviral remnants. So in this, in case they ever get reactivated, so these ERVs that mice, so they do this study in mice. So they show that in mice, a big part of preventing their reactivation or like the infection by these ERVs is mediated by pre-existing IgM expressing B1 B cells that are already capable of recognizing these glycoproteins, not only on these ERVs, but also in other glycoproteins that are similar in other viruses. I think they also mentioned COVID and influenza. And that if you have so they use a mouse model in which if you remove B cells or if you remove some TLR, I think they mentioned TLR7 and TLR9. So if you have these cells that have this, that are not able to have this, and you kind of cross the mice enough to activate this particular loci that it can get activated, that you end up having a retroviral production in this mice that is not protected because there's this IgM pre-existing IgM repertoire does not exist. So it's very cool because I was not very familiar with these B1 B cells, and I thought it was so cool that they seem to kind of already give you a head start when it comes to antibody responses to commonly to patterns commonly found in pathogens. So they are recognizing very specific kind of glycosylation that is mostly found in viral glycoproteins and not so much on kind of endogenous proteins, and that it doesn't really react with endogenous glycoproteins, which is also kind of interesting because it would be kind of, I guess, problematic if it would recognize your own glycoproteins with the same affinity. So I think that was kind of the idea I wanted to show of this paper is you have this pre-existing repertoire from the germline, encoded by the germline and expressed by this very specific fetal liver-derived population of B cells that are there mostly in the peritoneum in the mice, and that they protect you against this time bomb that you have in your genome, or at least in the mice. And they also found some human antibodies recognizing this glycoproteins in particular.
So without this, we all die.
I guess. I don't know how it would work, because if you get this reactivation, yeah, probably.
All right. I don't have a good segue here. What I do know, though, is we're going to be speaking to Dr. Lili Yang at the University of California, Los Angeles in a moment. Before I get to that, We're excited to be partnering with the International Union of Immunological Societies once again for the IUIS 2025 Congress, coming to Vienna, Austria from August 17th to 22nd. Join us for a week dedicated to cutting-edge immunology, innovative research, and global collaboration. Early registration and abstract submissions open on November 15th at www.iuis2025.org. Don't miss the chance to be part of the action.
And now we are reaching the second part of our show, and I'm very happy to be joined by Dr. Lili Yang. She is Professor of Microbiology, Immunology, and Molecular Genetics at UCLA, University of California at Los Angeles. And her lab is super interesting, very like engineering, has so many research lines, it's hard to keep track. So we're going to have a lot of really cool stuff to talk about today. Dr. Yang, thank you so much for joining the Immunology Podcast today.
Thank you, Brenda. It's a great pleasure to be here.
Thank you for being here. All right, Brenda, it's engineered cells, so I guess you have to go first.
I mean, it's my bread and butter. It's very exciting. It's not only engineered cells, but like engineering for the cells. So maybe I will let Dr. Yang give us a little overview of how does she describe her research interests and what are the most exciting projects going on in her lab? And just as a little kind of a primer for our listeners.
Sure, yeah. My lab, we have a name, we call it Young Engineering Immunity Lab, kind of captured, in those words, what we are doing. And essentially, we are interested in engineer the human immune system to fight off, various kind of disease conditions. And talking about the immune system, that we all have it, and it's very important to keep our body healthy. And for instance, the COVID flu infection, all those, like our immune system, is protecting us about it. And of course, enhancement like a vaccination, right? You probably all received the vaccines, and those help to boost the immune response and protect us better. We're doing something similar as what vaccine is doing to us, but through, various kind of technologies, including the gene and stem cell engineering, technology, which I can get into a little bit deeper later. But the overall idea is that we would like to take advantage of this evolutionarily equipped, defense weapon like we all have embedded in our body, and we want it to work better, for certain conditions, and we are utilizing the various kind of advanced, technologies, developed by biologists, by engineers, and to make the new generation of the medicine. And those all captured in this concept called immunotherapy, like engineering the immune system to, create new therapy. We have some special, focus for the disease areas, like cancer, like autoimmune disease, like infectious disease. And so that's generally what we are working on. And as Brenda, you mentioned, it's kind of a diverse, specific topics we're working on, but all under this common theme. And the most exciting, research, I would say, currently ongoing, in my lab, like we are trying to develop this off-the-shelf immune cell therapy. And why off-the-shelf? Because that we want to create something that really can work like the just the medicine, right? Like it's available, pre-made, and will be ready for prescription, if a clinician, diagnose the patient and would like to recommend this treatment, it will be available. Like it do not need to be customized, like one by one, and this many, steep price and logistics for delivery very personalized treatment. We want the cell therapy become immune cell therapy, becoming, more available, and more ready to go, and being able to cover a more like a broader, base of the patients. And so basically that's the most, like I would consider to be one of the most, exciting direction we are working with.
I guess that maybe that's a very interesting topic that you mentioned is off-the-shelf T-cell therapies or like cell therapies. Maybe it was a good starting for our discussion, because when I think of off the shelf, I think like of a few characteristics that I think you guys are working on all of them. Like on the one hand, you want targets that are present across many patients, and I think that when you're using CARs, that's kind of you're targeting surface molecules that are independent of individual polymorphisms. So that's usually CARs are more universally applicable. On the other hand, you want cells then are able to resist rejection by the immune system of the patient. I think that's very important. So I think you're also something that you guys probably are working on. And what I think is very, very interesting, and I know for a fact that you have a few papers on this, is starting material, not being, mature cells, but, generating these therapies right from hematopoietic stem cells, or stem cells that allow you from one donor make, really large amounts of very high quality cells. Would you like to kind of comment on this, and maybe for our listeners, elaborate a little bit more on what are the main challenges around doing you want to have immunotherapy for the masses in a way, like industrial scale immunotherapy, but what are the real issues bringing this to the clinic and really like making it a staple of therapy for cancer or maybe other kinds of diseases, and what have been the main methods or the main solutions that your lab or you guys have been working on or you think are the future of off-the-shelf immunotherapy?
Yeah, Brenda, I think you really touched the most important part of our research, and thank you very much for bringing up those points. Let me think about how to address this. Immune cell therapy, in particular in the form of CAR-T cell therapy, which probably many people heard about, it really revolutionized the treatment of the blood cancer. There's a couple of blood cancer type. It's really like, CAR-T cell make a huge, difference for the treatment, the kind of benefit can give to a patient. So that's stimulate a lot of excitement, right? So think about like how to make this powerful therapy. Number one, can we give it to all the patients in need, right? So the current answer is no, we cannot give to all the patients in need. Why? Because the current CAR-T cell therapy, is, as I mentioned, is quite personalized treatment, and there's a fundamental reason behind that. So you take a leukemia patient, right? It's suitable for the CAR-T cell therapy. You need to go through a whole procedure. You need to hospitalize the patient first and collect the blood from that patient. And the blood need to be sent to a manufacturing site and manufacture the CAR-T cell product. And then that product will need to go back to the hospital and give to that particular patient. And so it's involved a lot of, logistic efforts, and not all the patients have access, to such a medical center, such kind of logistics, and it also comes with a very steep price. The current market price, 6, CAR-T cell therapy has been approved so far, and the price is around $300,000 per, treatment. So all that combined, it's just the limitation, and for the reason I mentioned, because it's really really like a demanding like a process. And also it also not, all the patient even have all those, access to all those available, therapy, and may not work for them. And their blood may not be very, a kind of suitable for making a good product, or the manufacturing process itself can be, successful or not successful for particular case. I mean, it's because it's case by case. And there's a waiting period of time, right? So like the manufacturer usually take weeks, like sometimes more than months, for a product to be made. And during this time, patient cannot get the, product, they just need to wait. And sometimes this waiting period of time can be quite, devastating, because the disease progress fast and you may miss the time window, for good treatment already. So all that combined really are kind of really ask for like whether it's an alternative, right? So we know that the CAR engineered on powerful immune cell like T cells, can really treat, those disease. And how can we provide it in a way that it's ready to go? We do not need to pre-screen the patient. We do not need to worry like one particular like blood draw, one particular manufacturer, is successful or unsuccessful, right? And we do not need that waiting period of time. And once a patient is diagnosed or recommended for the therapy, they can have it right away, right? There's no waiting period of time. So all of that, it's I think for all the immunologists like us, kind of industry, like working in this area, that's the ultimate goal. Like if we can do that, that will be, the most ideal. There are some fundamental, scientific questions need to be addressed before we are able to make such a product. Okay, one of the most fundamental reason is that why we need to treat patients one by one. The CAR-T cell work with this T cells. Okay, every one of us, I have T cells, Brenda, you have T cells, everybody have their own T cell. And those T cells are very carefully like generated and educated, by individuals' body. Like my T cell is developed and educated in a way really fit my use, okay, and Brenda use is fit your use, okay, and in a way it's kind of have some correlation with this MHC molecule, major histone, compatibility, molecule, which match they need to work together with T cells to make them useful, okay, and MHC molecule among human population, it's a huge diversity. It's polymorphic and almost nearly impossible to find two individuals have the identical pattern of the MHC molecule. Exception is identical twins, but that's a very rare case, right? So it's commonly because of the mismatch of those polymorphism of this MHC molecule. One person's T cell only can be safely used for that person. Okay, so if you transfer that a person's T cell to other, a kind of individuals, it may cause some very, harmful consequence called graft versus host disease, and oftentimes that can be lethal. And that is why it's strictly, to be personalized treatment for the current CAR-T therapy. So we want to, think about ways to overcome this critical limitation, because I mean, you cannot really make an off-the-shelf product for everyone, if you limited by this, biological, restriction. So what we are thinking about is that we want to look for other type. We have many different subtypes of the immune cells, and we are looking for those type of immune cell as powerful as T cells, but it's not restricted by the polymorphic MHC molecule, right? And some cells is suitable for carry a CAR molecule, still do the type of function the CAR T cell can do right now, but it's not limited to individual person anymore. And that subtype of cell, we found them, their name called unconventional T cells, just separated from the conventional T cells like regularly used to make CAR T cells. They are unconventional in a way that they are powerful cells. They can attack tumor cells, and they are suitable to carry, tumor targeting molecule like a chimeric antigen receptor CAR, and they do not cause the GVHD, side effect when cross transfer, because that they recognize something common, kind of between people. So the one example of that cell is called NKT cells, natural killer T cells. So my lab at UCLA, since I started my independent lab here from 2013, so almost like it's a little bit over a decade, let's call it a decade, right? So in the past decade, my lab has been focusing on studying those cells, and we figure out the biology, figure out the tumor cancer fighting potential, figure out how to engineer them, to equip with the tumor targeting CAR, make them more powerful, we figure out all those. And then we also need to resolve another question. So see, this type of cell is what we want, but how to make enough number of them, right? Suitable for the clinical use, suitable, has a commercial path in the future, delivered to patients. And so that come to the stem cell engineering concept, okay? So those cells I mentioned, the NKT cells, as good as we love them, they are good, powerful potential. They are very little number. If you try to isolate from a healthy donor blood and try to expand them into a cell product, it's quite demanding because we have very small number at the beginning to start with. So then we think, how about we go back to this really powerful stem cell, go all the way to the type of stem cell give rise to those T cells. The stem cell always have a bigger proliferating potential, kind of can expand themselves, also can produce the, progenitor cells. And then we identified that stem cell to be blood stem cells, another name called hematopoietic stem cell, HSC. Those blood stem cells, they regularly live in the bone marrow of adult human, okay? But they are also very enriched in cord blood. So when a baby is born, that the cord blood contain, quite a good number and very good quality of those blood stem cells. And fortunately, there's a quite mature, industry supply chain, called cord blood banking system. Well, individual, like a family, can choose to banking the children's, cord blood for future use, or many families, also kindly offered to donate it to the cord blood banking, and in case it can be useful, kind of for some application for, treating, people. So it's a industry supply chain available over there. And so we thinking that how about we isolate the blood stem cell from the cord blood, donor and the banking system. And we engineer the stem cell in a way to make them to become the type of cell, not a random cell, right? Become the type of cell we want, which means the NKT cells. And during this process, we can smartly integrate, some other engineering approach to allow the cells coming out, from the culture to have the CAR, have the other enhancement, molecules, make them a more powerful, durable, safer, cell therapy product at the end. So that is kind of, we published a series of paper, to tackle the individual like technology, like breakthroughs and report that. And I think the overall concept at a more most matured form is reported in this, just in May, we published a paper on Nature Biotechnology, capture this entire process and idea, right? So like we eventually report, like we developed this clinically guided method, like starting from the cord blood, donor and stem cells, and we through a engineering approach and a culture method, and really can generate good number, high quality, high purity of the CAR-NKT cells, and in a quite robust way. We tested multiple donors and multiple generations around of culture, and proved that this is a robust way and have a quite clear like translational path into clinical and into a commercial path in the future. To give you an idea about what we have achieved using this method, say from one single cord blood donor, we are able to generate a trillion number, of this CAR-NKT cell at the end at a very pure condition. So there's no need to purify, no need to, have the intermediate like operation, and so it's quite a robust and streamlined culture. At the end, we only need to collect everything coming out from the culture and then direct freeze them. Okay, so cryopreserved. And we proved that the cryopreserved product, can stay stable, like for quite a long period of time, like at least a year. Like we test, like we pick the cells from the frozen stock from various time and recover them, and they are as powerful, as they were before. So like it's trillion number of what does that mean? Trillion number 10 to the 12th number, of those CAR-NKT cell, according to the current successful CAR-T cell therapy dose that can equivalent to around like 1,000 to 10,000 dose, depending on the high or low dose. So yeah, we are quite excited about this, method and accomplishment, because that means it's totally durable, right? So like a cord blood bank, a supply chain that you can get the say one single cord blood donor, you are able, now to make 1,000, say, dose of the CAR-NKT cell therapy. And as mentioned, because there's no, polymorphic like limitation, so those cells can be available, to give to any, cancer patient, like suitable, for receive this particular CAR-NKT design, and that hopefully can drive down the cost dramatically, and also totally resolve the logistic, requirement and the accessibility, requirement for whether a patient will be able even to receive such a treatment. So, well, of course, this is just the first step, right? So, like what we have reported is preclinical, study and the proof of principle, everything. We are very excited to moving along the path for translation that we already scheduled a meeting with FDA. We're going to discuss with them in December sometime, about the clinical translation of this method, and hopefully after that we will be, able to move into clinical testing, quickly, and then we hope like it may become a possible promising, new treatment in the future.
So you were going to go to my next question, which is, given that you are donor derived, the FDA is particularly specific about sourcing that and the regulations around that and the testing and the GMP validation of it. So it sounds like you haven't had that conversation yet, but you're planning to. Is that correct? Cool. So then I'm gonna go to my other question, which is the other angle of your work is looking at new different antigens, or not antigens, different pathways. They're checkpoint pathways other than, the classic two. And so I'm wondering if you could talk a little bit about, your favorite of the new pathways that you've been going after that are for checkpoint inhibition.
Certainly, Jason. Yeah. So thank you for bringing up that second direction, in my lab's research, we are equally, excited about. And as you pointed out, we are trying to look for the new, drug target, for developing, expanding the current scope of the immune checkpoint therapy, right? So one example of the therapy is anti-PD-1 treatment and the CTLA-4 treatment. Those has been, again, dramatically change the way, like how cancer and many type of cancer has been treated nowadays. All of the currently approved immune checkpoint therapy, drugs, all targeting this so-called classical, immune, molecules, like what immune cell has been interacting with and what we as biologists, understanding about them. So my lab like to expand the scope of those molecules in a way, like looking a little bit beyond the immune system itself, right? So we're trying to look like the interface of the immune system and other, biological system. And one, such interface is between the immune system and the nerve system, okay? So interesting enough that if you look at molecular level, the nerve system we have and the immune system we have many overlapping, molecules. Some molecule play a role, like it expressed like protein expressed in the nerve system and also expressed in the immune system. It has been a fascinating, area. Like people have been curious for a while, like what do those molecules do? What do they do for nerve, system? What do they do for the immune system? And going back all the way, like during the embryonic development, like those two systems for a period of time, they come from the same layers of the development. So there's something links them together. There's something shared in common. Okay. So what we did is that we did a screening, study. We have a animal, tumor model, and we isolated the immune cells, like infiltrating to the tumor. And then we isolated the immune cell outside of the tumor. We also isolated the immune system from a tumor-free animal, okay? Then we compare the difference of the gene expression to see what specific, gene or molecule has been changed for the immune cell infiltrating into tumor. And we lay our eye, particularly in this interface, the molecule involved in both immune and the nerve system. And that led to some very interesting discovery. And one, most exciting discovery is that we find that there's a network regulating serotonin, synthesis and the degradation and the transportation. It's really highly, changed, like in the tumor infiltrating immune cells. Okay, what is a serotonin? Serotonin, it work the best known, it has many functions, but one of the best known function is in the brain. It's kind of the happiness, substance, for the brain. For more serotonin, like be more cheer up, all those. So the serotonological system in the brain regulates the emotion and the behavior, of people. That's very well known. And because of this important function, all the molecules involved in the serotonin, regulation in the brain have been so well studied, like has become, some of them are the most defined drug target. And for a group of the drug called antidepressant. Okay, so like the antidepressant molecule can target the various part of the serotonological system in the brain to cause accumulation of serotonin in the brain, a kind of, boost the, happiness, right? It's a happiness substance. So we find, surprisingly, in the tumor, this serotonergical system also exists. It's kind of linked to the anti-tumor T cells. They are, as well as it's a happy substance for the brain, serotonin is a happy substance for tumor fighting T cells. They like serotonin. You have more serotonin, those T cells get better, they function better, they behave better, they fight tumor, better. So we find through the years, we find like two key molecules, both again are the major drug target in the brain to derive the antidepressant drugs. One is called MAO-A, so monoamine oxidase A. And the drug targeted is called MAO-I, monoamine inhibitor. And that is the first major group of antidepressant put onto the market, around probably 15 years ago. And then, it's kind of a little bit fade away. Now it's usually the backup line, of the treatment because the uprising of the other, more popular antidepressant drugs. And one of the most popular antidepressant drug called SSRI, selective serotonin reuptake inhibitor. And I can give you a brand name that probably a kind of call attention. Prozac is belongs to the SSRI. Okay, so that target some a molecule, uptake molecule called SERT, serotonin, reuptake transporter. Okay, so SERT, and we find both MAO-A and SERT are highly upregulated in the tumor fighting T cells in the tumor. And T cells, in fact, they make their own serotonin and they use those molecules to regulate the availability of serotonin to themselves. And then as a way, like the established antidepressant drug can also work for the immunotherapy. So we tested the most commonly prescribed antidepressant drug targeting either MAO-I, or the SSRI, drugs, and we find many of those work really well to help boost the anti-tumor T cell immunity, and because they work in such a different mechanism as the current existing and immune checkpoint, drugs like anti-PD-1, anti-CTLA-4, and TIM-1. So they can work really in a synergistic way as the existing drug. So what does that mean? It actually, has a quite, kind of important indication because even the current existing immune checkpoint therapy has been very successful, but it's not working for all the cancers yet. And for certain type of cancer, it not work for all the patient yet. And for the patient responding to the treatment, they oftentimes suffer from relapse later on, right? So some new type of drug can target a quite different, pathway mechanism of the anti-tumor immune response and can work alone or work synergistically, with the existing one, have the hope to cover those disease type patient population or dealing with the relapse situation for the patient to respond at the beginning and not responding later on. So we are quite excited, and this is also falling in an interesting category called there's a possibility to repurpose some existing drug. If you want to develop a brand new, a small molecule drug, right? From the beginning to the end, it's a very lengthy, process. You need to chemically, make it, you need to develop it, manufacture everything, and you need to go through this entire regulation, process, right? To clinical trial, everything to prove safety, prove efficacy, eventually bring it to the market. And that process, it's not short, and it's still, even nowadays, it has been lengthy, it will still be, quite lengthy. It's probably around 10 years, you bring something brand new, to the market. Repurposing an existing drug is different because you have it already over there, the safety. I mean, it's just like purposing the application of the drug. So that's significantly shortened the period of time, before it may be available for a certain kind of treatment. So this fall in that category. So we are equally excited for that part.
Yeah, I mean, that's really, really important, right? To be able to repurpose drugs already in the market. And, a few weeks ago, we also discussed another neuropeptides, neuropeptides that were actually, for example, able of influencing, Th1 versus Th2 responses. And it's very interesting to see there's so much overlap between these two different systems. So it's very exciting to see that it also has, you're finding in a very specific application for immunotherapy that could synergize with pre-existing therapies. We are reaching the end of our conversation. It's been very interesting to listen, to hear the research in your lab. And it was really, I think, especially, I also have some background in GMP manufacture and understanding all the sometimes feel kind of trivial or feel kind of unexpected challenges of doing cell therapies. This is what you mentioned, getting the cells in the first place. Patients with cancer usually don't have the best cells, the best starting materials. How do we work around that? How do we minimize all the logistics of transporting cellular products, which is a lot of work and a huge expense, and they cannot be delayed. They have to be, cryopreserved and transported across the Atlantic, maybe. So, what you mentioned really touched upon a lot of these logistical and kind of really fundamental issues with making cellular therapies available for all patients. So by the end of our conversation, we'd like to ask our guests a little bit of a question to get to know them a bit better, so for listeners to know a little bit their interests. So we call it the fun question. So I hope we have fun with it today. What I would like to ask you, what is the biggest misconception about science that you would like to correct and share with our listeners? Your response.
Yeah. So I think to me, I kind of learned along the years and correct it. I would like to teach the same thing to my student and maybe share with the audience today. So biological system is not perfect. We oftentimes try to think it's perfect. It's so delicate a design, right? It must be perfect. I mean, it's working. It's robust, but it's not perfect. There's tons of a lot of the redundancy, there's a lot of defects, of it. And so that also means it offers a lot of engineering opportunities, right? So there's many things we still need to learn from biological system. Again, it's such a complex system, amazingly robust and working, in many ways. And the more we learn, we can figure out what is the point, still can be improved further, and utilizing the more and more advanced technology like we are having nowadays, and the opportunity to make it work better.
All right. Well, thank you so much for coming on the podcast. It was a pleasure.
Thank you, Jason. Thank you, Brenda. It's a great pleasure to be here. Thank you for inviting me.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us at X at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.